Okay, hello you guys. We're going to go ahead and continue on in the 3.2 notes, talking about cellular respiration. In this video, we're going to look at topic 3, which is oxidative phosphorylation. And so this is going to be the last part of cellular respiration. So up to this point, we've talked about the first kind of few steps in cell respiration. We talked about glycolysis, which involves glucose breaking down into pyruvate in the cytoplasm of the cell. And then that pyruvate is moved to the mitochondria where pyruvate oxidation takes place. And that pyruvate is turned into acetyl-CoA. And then the acetyl-CoA in the mitochondria is going to enter the Krebs cycle or the citric acid cycle. And through those three phases, or those three different parts of cell respiration, all of that, by that time, the glucose has now been broken down completely into basically CO2 molecules. And so by the time you've gone through glycolysis and pyruvate oxidation in the Krebs cycle, we have now taken this 6-carbon molecule of glucose, and we've taken each of those carbons and they've now been broken apart into CO2 molecules that the cell is then going to get rid of. And during that process, we were taking away electrons little by little throughout this process where glucose was losing his electrons, and those were taken by all these NAD+ molecules swooping in, taking those high energy electrons that used to belong to glucose. And so by the end of this, those three parts there, we now have basically just these high energy electrons left unaccounted for. We need to know what happens to these high energy electrons, but the rest of glucose is now gone. We've broken it down, and all that's left is these high energy electrons. And so that's when we now enter kind of like the grand finale here of cellular respiration, which is oxidative phosphorylation. And so it says that during glycolysis, pyruvate oxidation, and the citric acid cycle, those things that we talked about in topic 2, there's a net of 4 ATP that have been made. 2 of those ATP, there was a net of 2 ATP made during glycolysis, and there was 2 ATP made during the 2 Krebs cycles that are taking place, starting with 1 glucose. And all of those ATP were made through a process called substrate level phosphorylation. But so far, that's actually not a lot of ATP from that 1 glucose. That 1 glucose has a lot more energy to offer, and all those high energy electrons are still holding on to a lot of that free energy that came from the glucose molecule. And we're going to tap into that energy to make a way larger amount of ATP during this last part here called oxidative phosphorylation. And during those processes, though, in topic 2, we stole a total of 12 pairs of high energy electrons. There was 10 NADHs that swooped in and are holding onto those high energy electrons, and there's 2 FADH2s that were swooped in and holding onto a pair of high energy electrons. You guys can go track them down in glycolysis, pyruvate oxidation, and the Krebs cycle, but there was 10 of these guys and 2 of these guys that we previously have created from that 1 glucose. And now they're going to take those electrons to the electron transport chain, where a lot more ATP is going to be made through this last part called oxidative phosphorylation. And this thing called oxidative phosphorylation, this occurs in 2 steps, the electron transport chain and chemiosmosis, which we're going to go ahead and dive into. And so we'll start with the electron transport chain. And so this is basically a series of protein complexes that are found in the inner membrane of the mitochondria. So if you guys look here, we have a cell membrane here with some proteins embedded in it. This membrane that we're looking at is the inner membrane of the mitochondria, which is, if you recall, here's the mitochondria. It has a double membrane. There's an outer membrane, and then there's an inner membrane that's all folded up. On this inner membrane, that's what we're looking at here in the electron transport chain. That's where we're going to find the electron transport chain, is on this inner membrane. And on one side of that inner membrane is this space right here where my mouse is pointing, the space between the outer membrane and the inner membrane. That's called the intermembrane space. And then on the other side of that membrane is this space inside called the matrix of the mitochondria. And so this membrane is separating those 2 spaces. But that's what we're looking at here with the electron transport chain. And that's where you're going to find all these different proteins that we're looking at right now. And so here's what happens. We got different protein complexes. There's 4 protein complexes that make up the electron transport chain. Protein complex 1, 2, 3, and 4. They are each identified by these Roman numerals. And this is where NADH and FADH2 are going to drop off those high energy electrons that they stole from glucose. So they used to belong to glucose at one point, and now they snatched those electrons and they're taking them here. And so what's going to happen is that these electrons are going to travel through these proteins. They're going to be passed along from protein complex to protein complex. And as they do that, their free energy is going to drop. So these electrons, these were high energy electrons that were taken from glucose, which means that they have a lot of free energy in these electrons. And as they're traveling through these protein complexes, what we're going to see is that the free energy that the electrons have is going to drop, and they're going to quickly become low energy electrons as they travel through here. And the energy from those electrons is going to be used to accomplish something here in the electron transport chain, which is what we're going to look at. So basically, those electrons being dropped off, which are represented by these little yellow stars right here. So here's NADH, he's dropping off a pair of electrons right here. And when he drops off a pair of those electrons at protein complex 1, he becomes, he goes back to being NAD+, and then he can go back and swipe some more electrons from glucose. But then those electrons, they're going to arrive here at protein complex 1, and energy from those electrons is going to be used to pump a hydrogen ion across this membrane into the intermembrane space. If you guys look at what's going on in this picture, protein complex 1 is moving a hydrogen ion from the mitochondrial matrix to the intermembrane space. And this is actually going against its gradient. In the intermembrane space, there's actually already a really high concentration of hydrogen ions. There's a lot of hydrogen ions out here. And in the matrix, there's actually a low concentration of hydrogen ions. So what we're doing here, protein complex 1 is moving hydrogen ions against its gradient, which is active transport. That requires energy to do that. That's not something that spontaneously happens. This is an endergonic thing happening. The universe doesn't want this to happen. And so this requires an input of energy. And protein complex 1 is actually using the energy from these electrons, these high energy electrons, to do this movement, this active transport of these hydrogen ions. And then once he takes some of the energy from those electrons and uses it to move a hydrogen ion, those electrons then get passed along. So then they go to this guy called the ubiquitin, which is just a helpful little protein here, who then carries those electrons to protein complex 3. And then protein complex 3 is going to use those same electrons and take a little bit more of their energy to pump another hydrogen ion out into the intermembrane space against its gradient. And then those electrons now have even less energy, and they're going to be passed along to this cytochrome C protein, which is another helper protein here, who's going to pass them along to protein complex 4. And protein complex 4 is going to steal even more of their energy, which they only have a little bit left at this point. They're going to steal even more energy from those electrons, those same electrons that made it through protein complex 1 and protein complex 3, and now arrive at protein complex 4. It's going to use some of their energy to pump another hydrogen ion out into the intermembrane space against its gradient. And by the time those electrons are through here, protein complex 1, 3, and 4, they have now gone from very high energy electrons that had a lot of energy. And now, as their energy has been taken away and applied to do this active transport, by the time they get to protein complex 4 here, they're very low energy now. These electrons have very little energy, very, very low energy electrons. And at this point, they're useless. All their energy is gone. There's not a lot of energy left to tap into. And so protein complex 4 needs to do something with these electrons. They need to go somewhere, because if they stay here, they're going to create a traffic jam, because what's going to happen is more and more electrons are going to always continually be dropped off. And so something needs to happen to these low energy electrons once they're useless and their energy's already been tapped into. And so what happens here at the very, very end of this electron transport chain is there's going to be a person who's going to be the final acceptor of these electrons. These electrons have gone through a long journey. They started in glucose and in the food that you were eating. They were taken either during glycolysis or pyruvate oxidation of the Krebs cycle by NAD+, swooped in and took these electrons. He carried them all the way here to the electron transport chain. They got dropped off. They got passed along, and their energy was taken away from them. And now someone's got to end up with them. At the very end, we got to put these electrons somewhere. And the final person to accept those electrons is actually going to be oxygen. Oxygen is going to be the final person to accept these electrons at the end of all of this. when no one wants them anymore, oxygen is going to take them. Which, if you remember in topic 1 of these notes, we talked about how these electrons will be taken from glucose, and ultimately during cell respiration, they will end up with oxygen. Oxygen will be the final acceptor of these electrons. Oxygen is going to be reduced. It's going to gain those electrons. We call that reduction. And when he does gain those electrons, he becomes water. And so that's what's happening right here. An oxygen atom, what this is trying to show you is there's an oxygen atom, a single atom of oxygen. It's kind of confusing here because it shows you 1/2 O2. Oxygen exists as a diatomic molecule, so it always exists as O2, two oxygens always paired together. But really, it's just one of those oxygen, just one oxygen atom in this oxygen molecule is going to accept this pair of electrons. And so every pair of electrons is going to be given to one of these oxygen atoms. And when that oxygen takes that pair of electrons, it becomes water. And that's what it's showing you here. It's showing you, okay, one of these oxygen atoms, along with 2 random hydrogen ions that are floating around, are going to come together and take these electrons and become a water molecule, is what's happening here. And then the other oxygen atom, well, he'll just wait for the next pair of electrons. They're going to endlessly be traveling and dropped off in this electron transport chain. And he'll become a water. But every oxygen atom will take those electrons and become a water molecule. That's what's happening here at the end. If you look, though, there's one difference here between the electrons that NADH drops off and the electrons that FADH2 drops off. The electrons that FADH2 drops off are going to be dropped off at protein complex 2, not protein complex 1. So here's the big difference. And those electrons, those high energy electrons that he's dropping off, they're going to skip protein complex 1, but they're going to be passed along to the other ones. So they'll be passed along from protein complex 2 to ubiquitin. He'll pass them along to protein complex 3, and then energy from those electrons will be used to pump a hydrogen ion out. And then they'll be passed along eventually to protein complex 4, and energy from those electrons will be used to pump a hydrogen ion out before it's finally given to oxygen at the end. So for that reason, the electrons that FADH2 drops off, they're actually going to pump less hydrogen ions out than the electrons that are dropped off by NADH. So these electrons from NADH are going to result in 3 hydrogen ions being pumped out before it's given to oxygen, whereas from FADH2, when they're dropped off here at protein complex 2, skipping protein complex 1, and protein complex 2 is not a proton pump, it doesn't pump hydrogen ions, those electrons will only pump out 2 hydrogen ions before being given to oxygen. So that's the big difference between those electrons and these electrons. But either way, it's the same basic idea of what they're accomplishing here in the electron transport chain. And then this is why you guys are breathing oxygen right now. Again, this is a really important job, and this is oxygen's biggest job in your body, is just to be here waiting in the mitochondria to take all these electrons as they are finished being transported here. But that's the electron transport chain. If we do some math here, we add up all the numbers, there's a total of, from one glucose molecule, there's going to be a total of 12 electron pairs that are taken from that glucose molecule. And you guys can go back and look at glycolysis and pyruvate oxidation and the Krebs cycle, and you'll see that there's 12 times that FADH2 and NADH take electrons. There's actually going to be 10 NADHs that are produced and 2 FADH2s that are created during those first few steps of cell respiration. And so that's 12 pairs of electrons total that were stolen from that one glucose molecule that we started with, which means that there's going to be 12 pairs of electrons dropped off here by NADH, those 10 NADHs and 2 FADH2s. So there's going to be 12 electron pairs traveling through this at some point, pumping out a bunch of hydrogen ions, which means you're going to need 12 oxygen atoms to collect each of those 12 pairs of electrons. So every pair of electrons has to be collected and accepted by an oxygen atom. So you're going to need 12 oxygen atoms, which would be 6 oxygen molecules. 6 O2 molecules would be 12 oxygen atoms, which would be all the oxygen you need to collect those 12 pairs of electrons after they're dropped off by these guys. And each one of those results in a water molecule being produced, H2O with a single oxygen. So that's going to create 12 water molecules, just to do the math here for you guys. And so those numbers are going to be important when we try to make sense of, when we try to add everything up and total what's going on, starting with 1 glucose. But I'll show you that at the end. But that's what's going on here. So that's the electron transport chain. Hope that basic mechanism takes place. And again, this is taking place on the inner membrane of the mitochondria. And we're producing a really high concentration of hydrogen ions here in the intermembrane space because of these guys, and a low concentration of hydrogen ions in the matrix. So then the question might become, what's the point of doing this? So why are we taking all these electrons from glucose, bringing them here? Why are we using their energy to pump these hydrogen ions to produce this concentration gradient across this membrane? Like, what's the point of this? And that leads us to this last, very, very last part called chemiosmosis. And so in chemiosmosis, we are now going to see why we're pumping these hydrogen ions out into the intermembrane space. So what we just did in the electron transport chain is we've created what's called a proton motive force. You guys might hear it explained like that sometimes. A proton motive force just means that there's a concentration gradient of hydrogen ions. Hydrogen ions, a lot of times we call them protons in biology. A hydrogen ion is just a proton. It's this positively charged small little atom that basically is just a proton. And so a proton motive force is just this concentration gradient of hydrogen ions that really want to go somewhere else, because there's a really high concentration, a really low concentration. So all these hydrogen ions really want to move back into the matrix. And so what's going to happen is there's a protein also on this inner membrane of the mitochondria. So the same membrane that the electron transport chain is located. There's another protein here. And this protein is called ATP synthase. And this protein will allow the hydrogen ions to go back down their concentration gradient back into the matrix of the mitochondria, from the inner membrane space to the mitochondrial matrix, from a high concentration to a low concentration, which is passive transport. So these hydrogen ions they really want to go back down their concentration gradient. And that's something the universe wants to happen too. That's exergonic. That's going to release free energy. We are accomplishing something the universe wants to do, getting closer to equilibrium. And what's going to happen is ATP synthase is going to use the energy from those hydrogen ions moving down their concentration gradient. That flow of hydrogen ions is actually a source of energy. There's energy that's being released when those hydrogen ions are flowing back down their concentration gradient. And it's going to use that energy to build ATP, to synthesize ATP, like the name suggests. This guy's called ATP synthase. His job is to synthesize ATP. And he's going to take ADP molecules. He's going to find ADP molecules and inorganic phosphates, which are just these phosphate groups floating around in the cell. And he's going to join them together to make ATP, which is something that's very endergonic. The universe doesn't want this to happen. It doesn't want ATP to be made. But this guy will use the energy released from these hydrogen ions going down their concentration gradient to power the production of ATP. That's what's happening here. So that's called chemiosmosis, that process of using a hydrogen ion gradient to power the production of ATP. We call that chemiosmosis. And so theoretically, and this is where the math gets a little sketchy because this is not totally accurate, but for the purpose of this class, it makes it simple enough to be true enough to think through until you learn that I'm lying to you a little bit. But so technically, well, not technically, this is kind of a lie, but we're going to pretend it's true. Each hydrogen ion traveling down its concentration gradient is enough energy to make 1 ATP. And so for every hydrogen ion that goes back down its concentration gradient, it's going to power the production of 1 ATP molecule. That's how we're going to simplify this a little bit, which then means that every NADH that dropped off electrons earlier in the electron transport chain, every NADH pumped out 3 hydrogen ions. And at some point, those hydrogen ions are going to go back down their concentration gradient through ATP synthase. And that could result in the production of 3 ATP molecules for every NADH that dropped off electrons. And then FADH2, it's a little different because he only pumps out 2 hydrogen ions or 2 protons. at the electron transport chain. And sooner or later, those two hydrogen ions that his electrons powered the active transport of out into the inner membrane space, sooner or later, those hydrogen ions are going to go back down their concentration gradient back into the matrix of the mitochondria through ATP synthase, and that can power the production of 2 ATP. So for every NADH, those electrons dropped off can ultimately allow 3 ATPs to be made. And for every FADH2, there's going to be 2 ATP that can be made for each pair of electrons dropped off by FADH2. And so those are the theoretical numbers that are kind of close to being true. And so if you think about it, this is going to add up to a lot of ATP because if we add that all up, we said that there was 10 NADHs throughout glycolysis, pyruvate oxidation, and the Krebs cycle that were created. And each of them is going to drop off a pair of electrons, and each of those electrons that are dropped off can pump out 3 hydrogen ions, and those 3 hydrogen ions can then lead to the production of 3 ATP. So for every NADH, you're ultimately creating 3 ATP from that. And so that would be 30 ATP from those 10 NADHs. I'm losing my spot here. Those 10 NADHs can lead to 30 ATP being created during the electron transport chain and chemiosmosis. And there was 2 FADH2s created during the Krebs cycle. The 2 Krebs cycles creates 2 FADH2s. And those will pump out 2 H pluses each. And each of those H pluses will result in 2 ATP being made. And so that would be a total of 4 ATP from those 2 FADH2s. So 30 ATP from these NADHs and 4 ATP from the FADH2. That's 34 more ATP molecules we were able to produce from all these high energy electrons that we stole earlier. So that's a lot of ATP that we're making. And so during this process of chemiosmosis and the electron transport chain, this is where the huge majority of ATP is made during cell respiration. The other steps of cell respiration, they didn't really make a lot of ATP. We made a couple in glycolysis, a couple in the Krebs cycle, but really this is where we're cashing in here during the electron transport chain and chemiosmosis. Now, these 2 steps, the electron transport chain and chemiosmosis, these together we call it oxidative phosphorylation. So you might be wondering, why is this called oxidative phosphorylation? So this whole process of the electron transport chain taking place, which is going to pump out hydrogen ions to then power chemiosmosis to take place to produce ATP, we call that oxidative phosphorylation, which is a different way or a different technique to making ATP, because what we're doing here, this is the phosphorylation part, where ADP is being phosphorylated to make ATP. And the oxidative part was these guys losing their electrons. So NADH losing his electrons, that's oxidation. FADH2 losing his electrons, that's oxidation. So them losing their electrons leading to the powering of this guy to phosphorylate ATP, to phosphorylate ADP into ATP, we call that oxidative phosphorylation. So we're using this oxidation process to power the phosphorylation of making ATP, which is different than substrate level phosphorylation, which was a different way of making ATP during glycolysis and the Krebs cycle. Or just memorize that these are 2 different things going on. But when I say oxidative phosphorylation, I'm talking about the ATP made right now during chemiosmosis and the electron transport chain working together here. So I hope that all makes sense. In class, we're going to diagram a lot of this to keep track of what's going on and to start putting this in our heads correctly. The only other thing on topic 3 of these notes is the fact that there's something called decoupling that can take place. And so there's this process that can take place sometimes in certain organisms where we decouple oxidative phosphorylation from the electron transport chain, meaning that we're going to disconnect the production of ATP from what the electron transport chain is doing, where we will allow these hydrogen ions to be pumped out by the electron transport chain into the inner membrane space, like this is showing here. But there's a way where we can let those hydrogen ions go back into the cell, or sorry, back into the matrix of the mitochondria without producing ATP. So if you guys look here, there's these things called uncoupling proteins. These uncoupling proteins are proteins embedded on the inner membrane of the mitochondria, just like the rest of these guys. And they actually let the hydrogen ions sneak back into the matrix without powering the production of ATP. So instead of going through ATP synthase, they can go through these uncoupling proteins, which sometimes we call these proton leaks because they're letting these protons leak back into the matrix. That's what decoupling means. And so that's going to result in less ATP being made. So if we have these guys in the cell membrane, in the membrane here, the inner membrane of the mitochondria, that's going to allow all these hydrogen ions to be pumped out because of all these electrons being dropped off. But then it's not going to result in as much ATP being made if some of them can cheat and get back into the matrix without powering the production of ATP. You're going to end up making less ATP. So this might seem not ideal. You might be thinking, like, why is this a thing? Like, why is he here? Because that seems to mess things up. Because now we're going to make less ATP from all those electrons that we stole, pumping out all these hydrogen ions. And this is actually something that certain organisms do to allow their metabolisms to continue producing heat without resulting in a bunch of ATP being made that's not being used. And so, like for endotherms, we talked about endotherms already. Endotherms are warm-blooded organisms who have kind of high body temperatures. And we're using our metabolism and the heat from our metabolism to control our body temperatures. And sometimes our metabolism needs to go faster, not because we need ATP, but because we need heat. And so I could be just sitting outside in the cold. And when I'm sitting outside in the cold, I'm not using a lot of energy. It's not like I'm running a marathon or anything like that. I'm just sitting down, but it's really cold outside. And my body's losing a lot of heat. And so to keep your metabolism going, even though your body doesn't need ATP, you have to trick your body to continue metabolizing things, to continue doing cell respiration, even though you don't need the ATP. So to trick your body, we have these uncoupling proteins that allow your cell respiration to continue going, which produces a lot of heat via your metabolism, without producing a lot of ATP. So then your body thinks that, okay, now we need to continue doing cell respiration. We're not making a lot of ATP. And so it tricks your metabolism and cell respiration to continue going more fast without a buildup of ATP. Because what happens is when your body starts making too much ATP and your cells aren't using it, it's going to slow down your metabolism. It's going to think like, oh, wait, we're wasting energy right now. We should slow down cell respiration. Like, why are we doing all this cell respiration if there's all this ATP that no one's using? And so these uncoupling proteins help trick your body. They help trick the body into thinking it's not doing as much cell respiration as it thinks it's doing. So just to summarize it, it's basically just allowing cell respiration to continue happening, which will result in heat being produced without a lot of ATP being made. Which is ideal for endotherms, especially when they need to continue making a lot of heat if their bodies are losing a lot of heat, even though they don't need a lot of ATP. This is going to help accomplish that. So that's what decoupling is when it comes to cell respiration, and why it's actually technically sometimes beneficial for this to happen, so that we can continue having that heat from our metabolism without making a lot of ATP when it's strategic to do that. But that's it for topic 3 here. This picture at the end gives you a nice summary of everything we just talked about with the electron transport chain and chemiosmosis, and how this is pumping out hydrogen ions and resulting in ATP being produced through chemiosmosis and ATP synthase. And then this very last slide gives you like a schematic overview of all these molecules that we're making, which is actually a really good picture to kind of start thinking through. If you guys look at this, it shows you we're starting with glucose, and in glycolysis, we're creating 2-pyruvate. And in pyruvate oxidation, we're creating 2-acetyl-CoA, which will allow the citric acid cycle to happen twice. And then over here, it keeps track of what's being made. So in glycolysis, you're making a net of 2 ATP and 2 NADHs. And in pyruvate oxidation, you're making 2 NADHs and 2 CO2s. And in the 2 Krebs cycles that take place, you're making 2 ATP, 6 NADHs, 2 FADH2s, and 4 CO2s. This is keeping a total of all of that. And then those NADHs and FADH2s, if you guys look, there's 6 plus 2 plus 2, that's 10 NADHs and 2 FADH2s. They're going to go bring their electrons to the electron transport chain in chemiosmosis. And in this process of oxidative phosphorylation, each of those NADHs will result in 3 ATP being made. So that's 6 ATP here. And those 2 NADHs, when they bring their electrons to the electron transport chain, that will result in 6 ATP being made. And those 6 NADHs bringing all their electrons to the electron transport chain will result in 18 ATP. And these guys bringing their electrons will result in 4 ATP. And so if you add this all up, that's what this is doing right here. And you'll see that there's certain things that need to go into cell respiration, like a glucose molecule. You're going to need that. You're going to need 6 oxygen molecules because you're going to have 12 pairs of electrons from these guys being dropped off, and each of those needs to accept those electrons, and each of those oxygen atoms accepting those electrons results in 12 water molecules being produced. But there's also some water that's needed for the citric acid cycle. And so if you keep track of it down here, you're going to need 6 water molecules to do the citric acid cycle twice, 3 per citric acid cycle. You're going to need 1 glucose to start glycolysis with, and you're going to need 6 oxygen molecules to collect all those electrons at the end. And you're going to end up making 6 CO2s right here and right here, and 12 waters right here during oxidative phosphorylation at the end of the electron transport chain, and a total of 38 ATP, theoretically, that you can get up to if all this happens perfectly. And so if I summarize that, the waters will actually cancel each other out. So overall, you use 6 waters but made 12 waters. So that's an overall production of 6 water molecules because you already used 6 to start this whole thing, and then you made 12. And so here's the overall formula for cell respiration. You have a glucose and these 6 oxygens producing 6 CO2s and 6 waters because you made 12 but used 6, and a total of up to 38 ATP, which this right here is the formula that you've learned since middle school. And now hopefully you can think through where all these pieces are and where all of this is actually happening, at least overall in detail what's going on in these different steps that we went through in topic 2 and 3. And then this number of ATP, it says 38 ATP. Just know that that is very theoretical. It's technically not 38 ATP. That's kind of oversimplifying this. The real answer is that if you had 1 glucose, you can make up to like 30 to 32 ATP. It's usually around there. And then there's reasons why it's not 38, but for the purpose of this class, we're not going to get into it. Technically, no one's going to ask you, like, what's the maximum ATP that can be made during cell respiration? Because 38 is technically not a correct answer, but it's kind of the answer we teach you first in these lower level biology courses in college, just to simplify things. But that's the gist. And so anyway, that's a lot of stuff. Cell respiration, that's a lot of stuff. But I hope a lot of that makes sense after watching these videos. Anyway, I'll see you guys later. Please make sure you watch and continue on to the topic 4 and topic 5 video at some point. But that's it.